end by thanking Gilles for having the foresight to move to Barcelona 5 years ago so you could host this meeting in the delightful city that we've had. Not only is the city absolutely gorgeous and on the seaside, but at the reception, the ability to look over the city and enjoy the scenery was phenomenal. Certainly, adding to that is some of the phenomenal architecture that you have from Gaudi, and including the ones that served as a model for the Star Wars stormtroopers. And it was really great. I noticed that, although I don't know how to use a pointer here, but if you look right on the top of the spires, there's the HIV model that Gaudi put together. I don't know if you can see it from your seats or not, but it's the unprocessed gags. So he clearly had the immature particles. I actually think his model, if you look at it carefully, is as good as any I saw today. In addition, you can tell that he used that's not an HIV model. If it was, the number of spikes would be much less. He used the SIV that had the transmembrane mutation, and as we know, that has about tenfold more spikes than the HIV does. So not only was he way ahead of his time in modeling the HIV, but he had this I find incredibly interesting, the way he modeled the cathedral itself. And I must say, Jeff, it's far superior to the model that you had for the HIV spike back in the 2005 Nucleocapsid meeting. But you know, there is a trimer there, and it's not too far off. So, Jeff is absolutely correct. We got into nucleocapsid studies, and it was the results of Lou Henderson early on purifying proteins of retroviruses and sequencing those and comparing them. And in this manuscript he published in 1981, in the abstract, if you look at it, he used the structure of Rauscher P10, and he said it's functionally homologous protein to Raus avian sarcomavirus, and the amino acid sequence homologies show they had a periodic placement of 3 cysteine residues in the GlyHis sequence, and they were found once in the murine protein and twice in the avian protein. Of course, this is the precursor for the zinc fingers. As you can see, this is the sequence that Lou was talking about with the CCHCs, and we know now that the question that was asked at that time, with that level of conservation, would suggest the nucleocapsid protein performs essential functions in the life cycle of the virus. The question was, what were those functions? And second is, could the nucleocapsid protein be targeted for drug and vaccine development? And that led us, and of course, after watching the presentations at the symposium, many people into fruitful areas of research. We know that the cysteines and histine coordinated zinc, and the wild type virus had zinc coordinated, and if it did, it packaged 100% of its wild type RNA and had wild type replication. One of the, for me, an eye-opening experiment was one that Rob Gorlick prepared, in which he would mutate any of the cysteine histidine residues, and if he replaced them with other amino acids, it ejected zinc. The virus packaging was eliminated, the zinc was eliminated, and there was no replication, obviously. But the other experiment that I was particularly impressed with that he did is when he replaced the histidine with cysteine so that he got zinc coordination, he had a functional molecule there, the CCCC. It packaged 100% of the wild type RNA, but it was replication defective. Ineffective to me, and I should have recognized that ahead of time, is that the virus nucleocapsid protein had a lot more functions than just packaging of the RNA. And after, again, listening to the talks in the symposium, it has many more functions than I would have ever recognized at that time, including virus assembly, RNA dimer maturation, reverse transcriptase, and integration. Now, as Jeff had said, back in 1998, we had been working on the nucleocapsid protein for a while, and I know that there were numbers of groups that were working on the nucleocapsid protein and felt it was important for us to have an opportunity to have these interactions. And this is actually the memo. Unfortunately, it's not on the letterhead, but I did print it out on the letterhead before I sent it out. It's a memo to our Associate Director of the Frederick Cancer Research Center, telling them that I'd like to hold a symposium at Frederick and ask if I could have some use of the facilities. And there I talked about the nucleocapsid protein being a replication process, also as a target for drug development. And in the drug development, Lou had done a fair number of work looking at different types of compounds that might be involved in injecting zinc. And that led us to, discovering mild oxidation would eject zinc. We then could use a compound that he had identified, alpha-thiol, to treat viruses. That would oxidize and activate infection of the internal portions of the virus and leave the envelope protein structurally and functionally intact, which in effect gave us a whole kill virus vaccine to be used in additional studies. When we did those vaccine studies, this vaccine was as good as any vaccine that had been used, including as good as one that Ron DeRocher had used of a attenuated virus. And as you know, the ways that we've stemmed virus epidemics is through vaccination, and most vaccines have either been whole particle or attenuated viruses. Neither one of those worked very well in preventing challenge by heterologous virus, which told us that HIV is going to be incredibly difficult. There are some new studies in which recombinant viruses expressing chronically envelope proteins might afford some help there. If you want to hear more about it, Jeff has been collaborating with people involved in that. So another thing on the memo I'd like to point out is that I had mentioned I had contacted two companies, Abbott and iGen. They had licensed our P7 capture assay, and they provided financing for it. The P7 capture assay was set up because, if you've been in the HIV field for a long time, you'll know that originally the way that HIV was measured was through a P24 capture assay. And if you tried to look for the virus in an infected individual, the antibodies to P24 competed with the assay. So for a long time, everyone thought that if a person was infected, there was a spike of infectivity, and then the virus disappeared until they developed AIDS and came back up again. Well, Jeff Lifson and Mike Piotok devised a quantitative assay for measuring virus RNA, and applied that assay to looking at plasma of HIV-infected individuals. And they showed in a seminal paper they published in 1995